Hello everyone and welcome back. Last time we got together we were talking about mitosis. Today we start meiosis or meiosis. No, it's meiosis. Alright. So up to this point now we should have a pretty good understanding of what mitosis is, right? So now we're going to take a look at meiosis. First off, first thing you need to know is meiosis has the same 4 stages as mitosis. Who can tell me what they are without looking at the board? Prophase, metaphase, anaphase, and telophase. Thank you. Yeah, we'll look at your worksheet. Nice. Now, wait, I heard it. How do you remember that? Pretty men always talk. Now, that's mitosis, right? Now, meiosis has these same phases, but it's going to go through them twice. No, not necessarily faster, but twice. Okay? So, if you have a cell that's going through meiosis, it starts out with growth, synthesizes, grows again, right? Remember that? In the P phase. No, what was that called? No, in the G1, S, and G2 phase. Also known as, what is that whole, all three of those together called? Interphase. Good job. All right. So, these same cells go through all that, but then when they get to where they're ready to split, they're going to split twice. Here we go. Let's figure out what's going on here. I'm feeling pretty crazy. So, the first stage of meiosis is prophase, just like it was in mitosis. But since meiosis is going to go through this stage twice, we give this stage a special name called Prophase I. Oh, you're getting ahead of me. All right. Who remembers what happens in Prophase I? Or who remembers what happens in prophase? Chromosomes start to condense. Chromosomes begin to condense. What else happens? There's four things, remember? Chromosomes begin to condense. The nuclear membrane starts to dissolve. What are those little things called? Centrosomes. Move to opposite sides of the cell. And what are they forming on those centrosomes? What are those lines called? Spindles. Yes. You guys are a little out of practice. We'll get back to it. Yeah. Chromosomes begin to condense. Nuclear membrane dissolves. Centrosomes move to opposite poles, and spindles form. So far, so good? Alright, and we call this Prophase I. Now, first thing, do you notice any differences between Prophase I and prophase of mitosis? Nope, they're identical. They're literally the same picture. So, no. I'm afraid there's no differences between prophase in mitosis and Prophase I in meiosis, other than the name. Okay, now, let me make sure I got this right. Oh, just kidding. Joke, I joked with you. There is one difference, and it's a major one. I almost blew it, and I'm sorry about that. Ladies and gentlemen, during mitosis and prophase, the chromosomes just condensed them, got wherever they got, lined up all willy-nilly. What you're going to notice in Prophase I, which you can kind of see in the picture, although it doesn't look much different than mitosis, is that homologous chromosomes are going to start pairing up. Now, what are homologous chromosomes again? Do you remember from before? They are similar, not identical. They're similar in shape, size, and kind. So, for example, in our little picture here, we have this tall blue one from, let's say, mom, this tall green one from, let's say, dad. Okay? And those two are very similar in shape, size, and kind. So how many of those would we have matching up, by the way? How many pairs? 23, right. 23 from mom, 23 from dad for a total of 46, or 23 pairs. I like to think of it as shoes. Got the left shoe from mom, right shoe from dad. You have 23 pairs of shoes. But you have 46 individual shoes. Making sense? Yes. The homologous chromosomes pair up. In mitosis, it didn't matter. Wherever the chromosomes, as soon as they condensed, wherever they ended up, they stayed. But in beginning parts of meiosis, they pair up. So in essence, if you looked at a cell in prophase 1, it'd have 23, a human cell, you'd have 23 pairs of dancing partners like. Making sense so far? Alright, so what? What do we care about? Do we even care? Oh yeah, oh we care. Oh we care. Ladies and gentlemen, because these chromosomes line up so close, it allows something to happen called crossing over. Okay. And this crossing over is what gives us some of our genetic variability. So, who knows what crossing over is? Yeah, no one does because we haven't talked about it yet, but guess what we're going to do now? Boom, a whole crossing over page. Here we go. Ready? What is this crossing over that Mr. Shab is talking about? Is he crazy? No. I'll tell you what he's talking about. Let's take a look. All right. First. Well, that's not first. That's the whole kit and caboodle. All right. First, we have us a pair of chromosomes, okay? Here's one from, would you want the green to be from mom or dad? Mom. Here's a green one from mom and a green one from dad. Now, remember, at the beginning of the cell's life, if you want, it would only had one chromatid, one side of this X, if you will. Okay? One from mom, one from dad. What happened in interphase during the S phase of interphase? Replicated. What was replicated? The chromatid or the DNA, right? So this chromatid was replicated right here. Notice the A's and B's are in the same spot. They are identical. So you have one complete copy here and one complete copy here. Same thing happened to dad's. You had one complete copy before the S phase. Now you have two complete copies. Are you feeling me? Okay. And these two are very similar. So what are they're similar in shape, size, and kind. So what do we call those? Homologous. Yes, homologous. All right. Now, what happens in crossing over? Well, each chromosome is made up of two sister chromatids attached with the centromere. See the yellow circle? By the way, what attaches to the centromere during cell division? Do you remember? I heard it. Spindle. The spindle fiber does. Excellent. And that's what pulls them apart? Yes, it's going to happen in meiosis also, but a little different. Okay, so crossing over happens when one arm of a chromatid crosses over the arm of the other chromatid. Notice, look, looks like, you know, green's tripping blue. Not very nice. But that's not what's happening. They are just so close together that the arms are getting mixed together. Remember, these arms are comprised of DNA wrapped around a histone disc, which is made into a chain, which is wrapped around a protein scaffold, which is tightly wound like a spring. Remember all that? Now, that tightly wound spring is mashing with another spring. So what's going to happen? Does blue chromosome fall over? Does green chromosome feel guilty? No. You know what happens? Crossing over. What will happen is each of these chromosomes is going to break at this point. Okay. Each of these chromosomes break at this point, and the green will attach to the blue, and the blue is going to attach to the green. Let us see. Maybe we're not going to... Oh, there it is. I knew we would see it eventually. Boom! Look what happened. All of a sudden, this chromatid, which it was a complete copy of mom, now has a little bit of dad in it. Oh, thank you. Oh, okay. Thank you. All right. And look at what happened to dad's chromatid. It now has a little bit of mom. Notice this one is still identical to the way it started. Notice that this one is identical to the way it started, right? But now these two are a little different. Now, are they a lot different? No, because remember these chromosomes are similar in shape, size, and kind, but they are changed somewhat. Okay, that is crossing over, and it only happens because these homologous chromosomes line up right next to each other. Had they never been near each other, no tripping would have taken place, no crossing over. Make sense? Questions? Yeah, this would only happen in prophase of meiosis, not in mitosis. Because in mitosis they don't get next to each other. All right. So what do you think is what comes after prophase in mitosis? Metaphase. And that is where we are going to tomorrow. Possibly it's metaphase 1. We'll find out. Thank you.